Happy Monday. How many Super Bowl hangovers are there today? So I'm happy to report the exam is finally ready. I just finished recording the grades about 10 minutes before class. So the exams are available in the Biochem office, ALS 2011. I don't have a grade distribution to show you yet because, as I said, I was just recording grades up until a few minutes ago. I can tell you that the low grade on the exam was 16, the high grade was 98, and the average was 72.5. That's a very good average, meaning that you guys have gotten on top of the material well. So I'm very pleased at the start. And as I think I probably told you last term, keep it going. What oftentimes happens is a class as a whole, I think, will feel like, oh, we've really got this together, and then the next exam they kind of fall. So let's avoid that happening this term. That would be great. So that's the word with that. Last time I finished up talking about the metabolism of propionic acid. And propionic acid arises when fatty acids with odd numbers of carbons are metabolized. As I said, they're not very common, but they are important to handle. So we don't want propionic acid accumulating. And the mechanism that I showed you, as I said, was an odd one. Start with a 3-carbon molecule, add a carbon in the wrong place, move it to another place, and then finally move it to another place. And the end product of that is succinyl-CoA. Succinyl-CoA, you know, of course, is an intermediate in the citric acid cycle. There is a vitamin that plays a very important role as a coenzyme in this process, and it's vitamin B12. That structure you should know for the next exam. Ha, 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 ha. Wouldn't you love to memorize that? It's a really hairy structure. It's the only place I'm aware of in biological systems where the atom cobalt is used. And cobalt plays an important role in helping to transfer a methyl group. And that's part of the reason that it's there. It's a very odd reaction, as you saw in the previous slide. And this is a very odd enzyme. Vitamin B12 is involved in other things as well, but this is one of the places where vitamin B12 is necessary. And we don't need to go through the mechanism. Okay, the peroxisome I mentioned briefly last time as being something that was necessary for the metabolism of long-chain fatty acids. And those long-chain fatty acids start their oxidation there as a result of an enzyme that's found only in the peroxisome, and that was the acyl-CoA dehydrogenase for long chains. And those longer chains typically are 18 or so longer, 16 or 18 longer. The peroxisomal oxidation scheme is different from the oxidation scheme that occurs in the mitochondrion. In fact, it's much less efficient. And the reason it's much less efficient is because there's not an electron transport system in the peroxisome. So those electrons that get removed during the oxidation process have really no place to go. So what the cell has evolved is this enzyme that actually takes the electrons off, puts them onto FADH2, that's the acyl-CoA dehydrogenase, and then that enzyme dumps the electrons onto oxygen to make hydrogen peroxide. So here is yet another place in the cell where hydrogen peroxide is being made. Hydrogen peroxide is a reactive oxygen species, so it's something that the cells don't want to have around for too much. Somebody last week sent me a message about their interest in limiting the production of hydrogen peroxide inside of cells. And I said that would be nice from the perspective of reducing reactive oxygen species. But unfortunately, cells actually use hydrogen peroxide as a signaling mechanism within them. So if you were to completely stop the production of hydrogen peroxide, you probably would have some significant impact. Because the electrons go onto oxygen and make hydrogen peroxide, they don't enter the electron transport system. And consequently, as I said earlier, the peroxisome oxidation is not nearly as efficient as it is in the mitochondria. Okay. The other thing I want to turn our attention to as we're moving in the direction of synthesizing things, which specifically we're talking about synthesizing fatty acids, is to discuss ketone bodies. Ketone bodies are sort of intermediate between the oxidation of fatty acids and the synthesis of fatty acids. And I'll show you why that's the case. So ketone bodies are important molecules. They're important anytime that our body is short of glucose. So anytime you're short of glucose for whatever reason, you might be diabetic, you might be starving yourself, you might have a variety of reasons why you're not getting, why your body doesn't have sufficient amounts of carbohydrates. The body needs a backup for the glucose that we use so commonly. Glucose, of course, travels easily in the bloodstream, and glucose is pretty important for our brain and for our eyes, neither of which are capable of synthesizing glucose. So if our body is not producing sufficient quantities of glucose for whatever reason, then if the body doesn't have a backup system, then what can happen is blood glucose levels can fall so low that one can have some significant problems. And in fact, I'm sure you probably have heard of people who've had hypoglycemia, for example, maybe fainted and so forth. To keep the brain alive and to keep the eyes functioning as much as possible, ketone bodies are that backup. And ketone bodies are important because they can be made from fatty acids. So fatty acids can't be used to make glucose, but fatty acids can be used to make ketone bodies. And I'm going to show you the process by which that occurs. If we look at the system on the screen, we see on the left two molecules of acetyl-CoA, one in red and one in blue. Okay? These would be the products of the breakdown of fatty acids. All right? In the synthesis of ketone bodies, those two guys are put together in the very first reaction here, going to the right, and that reaction is catalyzed by the enzyme thiolase, which was the enzyme that was the last enzyme of beta oxidation. Now, it might seem like thiolase is doing all kinds of things, but in fact, it's simply running the oxidation reaction backwards. That's all it's doing. If we had a 4-carbon molecule like this, and we were to metabolize it to the left, we would get 2 acetyl-CoAs using thiolase. So thiolase catalyzes this reaction backwards, and when it catalyzes the reaction backwards, we produce this 4-carbon intermediate. If we add yet another acetyl-CoA, so that we instead of having 4 carbons, we have 6 carbons, we produce a molecule that you've seen before. That's HMG-CoA. So HMG-CoA I mentioned briefly when I talked about cholesterol biosynthesis. HMG-CoA is a branch point between the synthesis of cholesterol and the synthesis of ketone bodies. So we're going to follow the ketone body path here. It's actually fairly short. After having put this 2-carbon piece on, 2 carbons are removed from it, and the 2 carbons removed yield a 4-carbon intermediate known as acetoacetate. Acetoacetate is a ketone body. It is a relatively unstable ketone body. Okay? Now, it's capable of traveling in our bloodstream, and in our bloodstream, when it travels, it can get to the brain, which might need it, and the brain says, okay, I need some energy. It will grab acetoacetate and run the reaction backwards, this direction, until finally it produces acetyl-CoAs. And acetyl-CoAs, of course, are useful for the citric acid cycle, and in that way, the body can keep the brain alive by producing acetyl-CoAs. Well, the other thing that acetoacetate can do, because it's unstable, is it can go and produce acetone spontaneously. This is not a reaction that requires an enzyme. This is simply a spontaneous reaction of acetoacetate. And acetone is not useful to the body. In fact, acetone is volatile. And so when it gets to your lungs by traveling in the bloodstream, you will breathe it out. And when you breathe it out, you will smell like acetone. If you ever smell acetone on somebody's breath, you should tell them they need to get to a doctor because it may be that they have diabetes. People who have diabetes and have problems with metabolizing glucose, in fact, produce acetone as a byproduct, and some people discover they are diabetic only after someone has recognized it on their breath. So if that happens, you should get them to a doctor. Almost broken my thing here. So that they can get taken care of. All right. Well, the other reaction that you see on the screen above is this reaction where acetoacetate is going up to D3-hydroxybutyrate. That turns out to be a protective mechanism. That is, D3-hydroxybutyrate is stabler than acetoacetate is, and so it can travel in the bloodstream fairly readily. It's not a ketone body technically. I mean, it's not a ketone technically, but people still refer to it as a ketone body. And that's because the reaction can readily be reversed to make acetoacetate, and then an acetoacetate can go back here. So the brain could just as easily grab the hydroxybutyrate and then run the entire process all the way backwards to making acetyl-CoA. Yes? Excuse me. The question is, does acetone smell more pungent if a person is on a low-carb diet or if they're diabetic? It's acetone in either case. So the question would be how much acetone is being produced, which might be part of what your question is asking. And the amount of acetone being produced would really be more of a function of how low their blood glucose is. So I would say you probably couldn't tell the difference between the two. Okay. Other questions? Yeah. I'm sorry? Yes, acetone will be excreted by breathing. Will it come out in sweat? Well, because it's a volatile compound, and because it's somewhat soluble in water, I would imagine it could come out in sweat. I would imagine it probably also could be released by the kidney. So you might have it come out in urine as well. Most commonly, it'll come out in the breath. Okay. Well, those are the ketone bodies. We don't need that. Utilizing it. So here's the reversal of that reaction I was telling you about. So we're going from acetoacetate down to acetyl-CoA, going backwards. And we'll not talk about that. Okay. Well, let's talk about fatty acid biosynthesis. Fatty acid biosynthesis is, as I noted in the lecture on Friday, very chemically similar to the reversal of fatty acid oxidation. There are some differences, but the chemistry of it is very similar to what you've already seen. One of the places in which fatty acid synthesis differs, and by the way, I'll remind you that fatty acid synthesis occurs in the cytoplasm primarily. Longer chain fatty acids, that is 18 or longer, are made in the endoplasmic reticulum and to a limited extent in the mitochondrion. And I'll talk about that in just a little bit later. Fatty acid biosynthesis starts in a different way than fatty acid oxidation ends. Fatty acid oxidation ends with acetyl-CoA, and those acetyl-CoAs are produced simply by chopping 2 carbon pieces off each time as beta oxidation proceeds. There's an additional step in going backwards and making fatty acids, and that additional step, is what you see on the screen. The additional step is an interesting and odd one also. All right, acetyl-CoA is the starting point, and acetyl-CoA is combined with bicarbonate, that is a source of a carboxyl group, and using ATP, it makes this molecule called malonyl-CoA. Now, malonyl-CoA has 3 carbons, not 2, and malonyl-CoA is in turn converted into something else, as we shall see. All right? The enzyme that catalyzes this reaction is the only enzyme in fatty acid biosynthesis that's regulated. It's known as acetyl-CoA carboxylase. Acetyl-CoA carboxylase. Now, carboxylases, you have learned previously, are enzymes that put carboxyl groups onto things. Here's the carboxyl group coming on, and you see it in red on the malonyl-CoA on the end. Okay? Acetyl-CoA carboxylase is regulated in two ways. It's regulated by covalent modification, and it's also feedback inhibited by one compound and activated by another compound. Okay? Let's talk about the feedback inhibition and the activation first. Okay? The feedback inhibition of acetyl-CoA is the end product of the synthesis of fatty acids. The first end product, there are others, but the first end product of the synthesis of fatty acids is palmitic acid, or palmitoyl-CoA. That is a fatty acid that has 16 carbons. That's why, and as I said last time, it's the most abundant fatty acid in our body. And the reason it's the most abundant is the first round of fatty acid biosynthesis stops there. And many fatty acids don't get any longer. They just stop at 16, and that's that. Palmitoyl-CoA / palmitic acid is a feedback inhibitor of acetyl-CoA carboxylase. When it binds, the enzyme will be turned off. Okay? Well, what's an activator of the enzyme? The activator of the enzyme is interesting. You can actually figure it out, and we're going to figure it out together. Let's imagine that you're a cell. And you're a cell that has that's residing in a person that's not exercising very much. And that person who's not exercising very much is sitting around watching the tube, eating beer, drinking beer, eating pizza, whatever it is that they do. These kids these days, I don't know. Drinking beer, eating pizza, and not getting much exercise. So my first question to you is, how much NAD do you suppose they have? High or low? They actually have low NAD. Okay? They have low NAD. Why? Well, let's think about this. How much ATP do they have? They have high or low? High ATP, what's happening to the proton gradient? Proton gradient is going to be high, right? Proton gradient's high. Cells have no place to put electrons. NAD is going to be low, right? So NADH is going to be high. NAD is going to be low. Let's now step backwards. We're in the mitochondrion still. Let's step backwards and think about the citric acid cycle. In the citric acid cycle, what's that? Oh, okay. In the citric acid cycle, all right, where's the first place that NAD is needed? It's in the isocitrate dehydrogenase reaction. That's the first place that NAD is needed. That means that isocitrate will accumulate. If isocitrate accumulates, what's the precursor of isocitrate? So, if isocitrate accumulates, citrate will accumulate, right? Turns out citrate is the allosteric activator of acetyl-CoA carboxylase. Now, when we think about this, we now see why we're making fatty acids when we're not when we're not exercising. We're making those fatty acids because the citrate is activating acetyl-CoA carboxylase. Now you're sitting here thinking, but wait, where is acetyl-CoA carboxylase? You said these reactions are occurring in the cytoplasm. Isn't citrate found in the mitochondrion? And if you said that, I would almost give you an A. All right? You're correct. Citrate is found in the mitochondria, and acetyl-CoA carboxylase is found in the cytoplasm. So there shouldn't be a problem, right? Well, it turns out, no, there actually is a problem. The problem is that we get citrate out into the cytoplasm, and we get it out to the cytoplasm as follows. Here is citrate accumulating. We've gone from oxaloacetate, forget the pyruvate, but we've gone from oxaloacetate. We've added an acetyl-CoA to make citrate, but now citrate isn't going any further. Citrate is starting to accumulate inside of the mitochondrion. When it accumulates inside the mitochondrion, it gets transported out of the mitochondrion, and you might wonder why. Well, the reason why is it's carrying on its back acetyl-CoA. Citrate is a shuttle for carrying acetyl-CoA out into the cytoplasm. It's a shuttle. So it comes out here, and the reaction that you saw here where it got added is simply reversed. and acetyl-CoA is produced. Now, we have a mechanism of delivering acetyl-CoA out of the cytoplasm, and it happens when citrate concentrations get high. Okay? So, now we see the connection with obesity and fatty acids as a result of not exercising. All right. So, what I told you, all right, so let me talk about the phosphorylation as well. Okay? So phosphorylation of the acetyl-CoA carboxylase has an interesting effect. Acetyl-CoA carboxylase exists in the cell as a long polymer. That is, hundreds and hundreds and hundreds of subunits are linked together. Okay? They're linked together. And they form these long strands that are actually found in the cytoplasm. You can actually see them in a light microscope, amazingly enough. They're very, very large. Okay? When the complex gets phosphorylated, that's the covalent modification that it gets, that polymer is converted into a monomer. And that conversion of the polymer into a monomer reduces its activity significantly. So phosphorylation inhibits the action of acetyl-CoA carboxylase. Dephosphorylation favors the formation of polymers. Okay? So the ideal system, if you want it most active, you would have it dephosphorylated and you would have citrate present. And if you want to have it the most inhibited, you would have it phosphorylated and palmitate present. Okay? All right. Question? Yep. Why is it taking it out? Okay? So it's going out because it's accumulated. So citrate is accumulating, and any excess of something is going to be removed in some way. Since it's not being broken down into other things in the citric acid cycle, it will be taken out.
So then the acetyl-CoA just interacts with the acetyl-CoA.
So once the acetyl-CoA makes it out into the cytoplasm, his question is, what happens to the acetyl-CoA? So the acetyl-CoA in the cytoplasm will interact with the acetyl-CoA carboxylase in favor of the formation of malonyl-CoA, and then we're often running to make fatty acids. Okay? Now, we're not quite there. There's a couple other things that actually have to happen that are different from fatty acid oxidation. Okay? One is the carrier. Out of the cytoplasm doesn't use CoA on molecules that are being synthesized into fatty acids. Instead, they use something called acyl carrier protein, or something I call ACP. So when we look at fatty acids being synthesized, they're not linked to CoAs, they're linked to ACPs. We're going to see that to start the synthesis, we're going to have one malonyl ACP, remember the malonyl CoA that we had, and we're going to need one acetyl ACP. So we need to have both of those things in order to start fatty acid synthesis. If we look at the side chain of the acyl carrier protein, we see that it has the same functional group that coenzyme A has, meaning that cells really like this handle. Why they use a protein instead of CoA, I can't tell you. But suffice it to say that the handle that's holding on to the fatty acid, the growing fatty acid, is exactly the same. Okay. All right. Now, here are the reactions that make things. Let's see, I thought I had an ACP. Don't have it. Okay. Well, actually, here, before I go further, here's the acetyl-CoA carboxylase. Those are actually the filaments of these guys that you can see. That's actually an electron micrograph, but they're very large filaments that form. Yes?
I just wanted to know if protein kinase is responsible for phosphorylating.
Question is protein kinase A responsible for phosphorylating? No, there are other kinases that can do that. Okay, But protein kinase A, I believe, may also be able to do that. Okay? All right. Let's see. I think I've got... Yeah. So here, it's called an AMP-activated protein kinase. All right? What is an AMP-activated protein kinase? Okay? Think about that. What does AMP tell in the cell? Low energy, right? Do you want to be making fatty acids when you have low energy? Nope. So you have a kinase that when it binds to AMP, activates it and causes it to phosphorylate the carboxylase, so the phosphorylase is inactive. Pretty cool. It makes sense, right? There's our friend phosphoprotein phosphatase, otherwise known as protein phosphatase, that's taking the phosphate off. When would we expect the phosphate to be taken off? When what hormone is active? Does anybody remember last term? Not epinephrine, it's the opposite one. Insulin, okay? Remember, what's insulin doing? Insulin says, we got all this glucose out there, bring it inside the cell. When all that glucose comes in the cell, what's the energy state of the cell? High, right? Energy state of the cell is high. You want to be storing energy. In this case, we're making fatty acids and storing energy. So that is consistent with things that you've already learned. Okay, now let's go to the reactions. So if we take an acetyl-CoA and we react it with an enzyme that puts an ACP on there, we end up with an acetyl-ACP. If we take a malonyl-CoA and we react it with the same enzyme, we will produce a malonyl-ACP. Now, I said we had to have one of each of those to start the process. Okay? One of each of those to start the process. Notice that the malonyl is in red and the acetyl is in blue. That'll help you to orient yourself as we go through this reaction. The first reaction is a condensation. Condensation is joining these two guys together. And interestingly, when we join them together, the CO2 that was put onto make the malonyl-CoA is removed. So this CO2, which was added onto acetyl-CoA to make malonyl-CoA, it's removed in the very first reaction. This is one of the reasons it's odd. We put it on, we take it off. Where have you seen a similar reaction of putting it on and taking it off before? Again, you saw it last term. Remember it? 2 points extra credit for who can tell me. Triad? No? Nobody? All right, maybe that'll be your extra credit question for the next exam. Okay? You've seen a reaction like that. You saw it last term. Okay? Where it got put on, and then after it got put on, the same thing got taken off. No, no. I can't, well, I'm not going to give you any hints. I'll let you guys look it up, okay? All right, so anyway, so this carbon dioxide gets put on, the carbon dioxide gets taken off, all right? ACP comes off because we've got two ACPs and we only need one. So now what we've done is we've attached the acetyl group onto the end of the ACP. All right? We've attached the fatty acid onto the end of the ACP. We've lost the carbon dioxide. All right. Now we have something that looks like the end of fatty acid oxidation. Now we're ready to start running those reactions backwards. Instead of oxidizing, the next step we're going to reduce. So we use an NADPH. Notice it's NADPH to reduce this carbonyl group down to an alcohol. That is a reduction reaction. This reduction reaction produces this guy here. One of the differences between fatty acid oxidation and fatty acid synthesis is the orientation of the hydroxyl. In fatty acid oxidation, the hydroxyl was in the L configuration on carbon number 3. In fatty acid synthesis, it's in the D configuration. We can tell the difference by that. Okay? Next, water is taken out of the molecule. Notice water is taken out. In the fatty acid oxidation reaction, water is added. We take water out of the molecule. That's a dehydration. By the way, dehydration means you take out water. Dehydrogenation means you take out hydrogen. Don't confuse those two. Okay? Dehydration versus dehydrogenation. They're not the same thing. Dehydration reaction produces something that's got a trans double bond, just like we saw in fatty acid oxidation. The last step in the process then is addition of protons and electrons from NADPH again to make this guy right here. So notice now we've gone from something that had an oxygen at position 3 down to something that has hydrogens at position 3. That is 2 sets of reductions that took us to get to that point. At this point, we have a 4-carbon fatty acid. This 4-carbon fatty acid will assume the position of the 2-carbon fatty acid up here. So it will combine with a new malonyl-CoA, and notice that the fatty acid will be put on the end. So the newest part of the synthesis will always be the most recent malonyl-CoA, malonyl ACP that came in. Okay? The newest part will always be the most recent malonyl ACP. All right. This process will continue until we get to a compound that has 16 carbons. That's palmitoyl-CoA. At that point, palmitoyl-CoA will be released. And the synthesis in the cytoplasm will be done. Yes? Why do we use NADPH? I think that's a question somebody asked the other day. The answer is that cells do tend to use NADPH for biosynthetic reactions and NAD for accepting electrons in oxidation reactions. Maybe it's just easier to keep track of. Okay, so the question is, do I know why, is there a chemical reason why it's NADPH instead of NADH? I don't think the charge has any significant effect on that, although you could imagine that the enzyme would might accept one. But the charge per se has nothing to do with the donation of the electrons or any of that. Okay. Yeah.
You want to just know all the names of intermediates or just steps?
Okay, good question. What do I want you to know out of this thing? Well, I haven't given you any names, right? So I haven't said any names here. I have talked about the general structures. And so I think, again, whatever I talk about is fair game. I haven't given you any enzyme names. And it turns out this is one of the easiest pathways you'll get for enzyme names. You know why? Because all of these activities that you see right here are contained in a complex. And each activity has its own name, but I say, why in the hay should we memorize every name of every one of those? And of course, the names are about this long. We should do what most people do. We should name the complex itself. And the complex has a very complicated name. It's called fatty acid synthase. Fatty acid synthase. Okay? So all these reactions are catalyzed within fatty acid synthase. Fatty acid synthase will synthesize fatty acids up to 16 carbons. If we want to go longer than 16 carbons, we have to have excuse me, other enzymes that will do that. Up to 16 carbons, fatty acid synthase will handle it just fine. Okay. There's the reaction summary. There's the names of all those enzymes over there, all contained within fatty acid synthase. Okay. This schematic, I find, is more confusing than it is helpful. So my advice would be probably to avoid this schematic. Let me tell you what fatty acid synthase does, though, that this figure at least tries to communicate to you. All right? Fatty acid synthase takes those ACPs linked to a growing fatty acid chain. And if you think about this, this guy has several reactions it has to do. All right? It has to do a condensation. It has to do a reduction. It has to do a dehydrogenation. It has to do, a hydrogenation, and we finally have a fatty acid. If we think about that, this is actually like a clock. All right? And it turns out this enzyme has a structure that does a rotation through these reactions. Okay? So if we start, let's say, down here, I have a reaction of a condensation. I move over here, I have a reaction of a reduction. I move over here, I have a dehydration. I move over here, I have a hydrogenation, and I'm back where I started, back down here. So this guy actually has a rotating arm, kind of like, it's not as fast or not as interesting as the arm that's involved in the ATP synthase, but it does have a circular design. This circular design turns out to be pretty much conserved through a good deal of biology. You can find it in bacterial cells, and you can find it in your cells. So it says that there's something about the efficiency of putting all these things into one place that allows cells to make these compounds. Okay, let's see. Control of acetyl-CoA carboxylase here. All right, now, the unsaturated fatty acids are also made from the saturated fatty acids. So notice what I've shown you so far is how to make the saturated fatty acids. Because each time we're going through two reductions and we're producing saturated fatty acids. All right? Saturated fatty acids are the precursors of the unsaturated fatty acids, not surprisingly. And the unsaturated fatty acids have electrons and protons removed from them to create cis double bonds. Okay? The enzymes that catalyze those reactions are known as desaturases, and they're found in the endoplasmic reticulum. My first question to you, based on what you've seen of fatty acid oxidation, which has similar reactions to what we're talking about here, what do you suppose the electron carrier is going to be for making an unsaturated fatty acid? NADP, I hear here. Other thoughts? FAD. FAD makes sense. Why does FAD make sense? Well, it's in the cytoplasm, but that's not the reason. There's a more chemical reason why it makes sense. Okay, so let's think back to fatty acid oxidation. The very first reaction, what was that reaction? It was a dehydrogenation. It was taking hydrogens and electrons from carbons that only had hydrogens on them. So if we're going to take a fatty acid and we're going to pull hydrogens and electrons from them, they're going to have to go on to the same electron carrier that we would use as if we were oxidizing them, because that desaturation is an oxidation, folks. It is an oxidation. So it's going to need to go on to FAD instead of NAD. Okay. The desaturases, as I noted, are located in the endoplasmic reticulum, and I mentioned earlier what the my mind's not working today. What the essential fatty acids are. Essential fatty acids are fatty acids that we have to have in our diet because we can't synthesize them, and as animals, we can't put double bonds farther out than delta 9. Okay? So the desaturases we have are limited in terms of where they can put those double bonds onto the fatty acids. Those, as I said, won't go past delta 9. Okay, so that's how we make the unsaturated fatty acids. If we look at the unsaturated fatty acids listed here, the only one of these that we can make in our bodies directly is palmitoleic acid, that guy right there. Everything else out there past delta 9, and this is the omega scheme, which is different, but all these other ones are past delta 9, and we can't make those. We have to have them in our diet. If we don't have them in our diet, we will be deficient in fatty acids that we need. Okay, questions about that? Okay. The last thing I'll mention about fatty acid synthesis, and then we'll do a song. The last thing I'll mention about fatty acid synthesis is the elongation of fatty acids. Fatty acids are produced in the endoplasmic reticulum and the mitochondrion. Now, mitochondrion is where oxidation is going on. Won't we have a futile cycle if we have synthesis of fatty acids going on in the mitochondrion when we have degradation of fatty acids going on in the mitochondrion? Well, it turns out no. Remember that the longer chain fatty acids, okay, are being oxidized not in the mitochondrion, but they're being oxidized in the peroxisome. That's where the acyl-CoA dehydrogenase is that works with the longer chain fatty acids. Right? So if we take a 16-carbon palmitate and we run it into the mitochondrion, what's going to happen is the elongation enzymes are going to grab it and they're going to make a longer chain fatty acid. They're going to start adding carbons to it. And they go through the reactions very much like what you've already seen here. I'm not going to distinguish them from what you've already seen. Okay? They grow 2 carbons at a time. All right? They don't have ACPs on there, but that's another matter. All right? They grow 2 carbons at a time, and they can end up being as long as about 26 carbons or so. All right? But because there's no enzyme in there that'll touch those in that longer state, they stay there, or at least they get made. They don't get oxidized. And then they can be used by the cell as appropriate after that. Now, the mitochondrion is a relatively minor way of making the longer chain fatty acids. They're more commonly made in the endoplasmic reticulum. And in the endoplasmic reticulum, we have the same setup that we had in the mitochondrion, adding 2 carbons at a time, doing very similar to the reversal of the fatty acid oxidation reactions to produce the longer chain fatty acids there. Okay. And those enzymes we categorize with a simple name, elongases. E-L-O-N-G-A-S-E-S. All right. We have finished fatty acid synthesis. Now, on Wednesday, I will talk more about arachidonate and prostaglandins. I'll probably get a little bit ahead, which is also good. But in the meantime, if you guys will sing loud, we have a song, and then I'll turn you loose. It's a tune you've already heard twice. Oh, what did I just do? Oh, man. Hold on. I thought I had it up there. It's called When Acids Are Synthesized. I want to hear you. The 16-carbon fatty acid palmitate gets all the carbons that it needs from acetate, which citric acid helps release from mitochondria matrices. Oh, a shuttle's great when acids are synthesized. Carboxylase takes substrate and it puts within dioxycarbon carried on a biotin. CoAs all gain a quick release replaced by larger ACPs, and it all begins when acids are synthesized. A malonate contributes to the growing chain. 2 carbons 7 times around again. For saturated acylates, there's lots of NADPH that you must obtain when acids are synthesized. Acetic acid made this way all gets released. Desaturases act to make omega-3s. The finished products, big and small, form esters with a glycerol. So you get obese when acids are synthesized. Okay, see you guys on Wednesday.